Hi everyone, Dr. Hatami is here. In this video, let's talk about mitosis. Mitosis is a type of cell division that our body needs in order to grow and maintain itself. During mitosis, one cell divides to produce more cells. So we need mitosis before birth and after birth for overall growth. During adult, we need them for to replace the old and damaged cells and also to help with repair after injury. Um, so as we discussed earlier in the cell cycle video, before a cell can enter the mitosis, it must go through interphase. During interphase, the cell grows larger, duplicates all organelles, and replicates its DNA. So by the time the cell enters mitosis, everything is already copied and the cell is fully prepared to divide. Now let's talk about the parent and daughter cells. So the original cells is called the parent cell, which produces two daughter cells. So we call them as daughter cells. They are genetically identical to each other. The daughter cells are totally identical from their genetic. So for example, if like one daughter cell has 46 chromosome, the other one also has 46 chromosome. And also daughter cells are genetically identical to the parent cell. So the parent cell also has this 46 chromosome. And also they are roughly the same size. What is happening during mitosis? Mitosis is actually a division of the nucleus. So during the mitosis, which it has 4 phases, we are going to divide the nucleus. So you should know these mitosis phases by order, which the first phase is prophase, and after prophase we have metaphase, then anaphase, and then telophase. So you should know them by order. First stage or first phase is prophase, and the last one is telophase, which we are going to divide the nucleus. Let's start with the prophase and try it together. During the prophase, the nuclear membrane disappears. So now let's show that in our picture by dash line. So this is our cell, and this is actually the like fragmented nucleus, or let's label this with our nuclear envelope, which as you can see is fragmented. Okay, so we'll show that chromatin has condensed to chromosome, and let's draw 4 of them. So now we are going to see the chromosome. So during the prophase, we are seeing the visible chromosome. So these are visible chromosome. So we know that in human cells we have 46 chromosome, but we just draw 4 of them because it's hard to draw 46, right? Okay, next we are going to show the centrioles, which during the prophase centrioles move to opposite poles of the cells. And remember they exist in pairs, and there's 2 pairs of them because they duplicated in interphase. Now let's draw the 2 pairs of centrioles. which they move to the opposite poles. And here we have our centrioles. Okay, and spindle fibers start to form, so they look like a little whiskers forming from the centrioles that will be anchoring them. So then here, let's show this spindle fiber, something like that. And also do not forget to label the spindle fibers. Okay, that is all happening through the prophase. So let's review it one more time. The prophase is the first stage of mitosis. We saw that how centrioles move apart to the opposite ends of the cells, and then the spindle fibers start to form from the centrioles, which they help separate chromosome. Then we saw how nuclear envelope begins to fragment, and then chromosomes condense, and that is the time that we are going to see the visible chromosomes, and then each chromosome has two identical sister part. So when we have this chromosome, we are going to have like each of these as a sister chromatid, and then which they would be joined at the center, which is called centromere. Centromere. So that is our chromosome. Okay, next phase we have a metaphase, which is the second phase of mitosis. And let's draw this large size circle to represent this cell. And then at this time, chromosomes are lining up in the middle, which this area is called metaphase plate. So let me show this like part of the cell in the middle as metaphase plate. The centrioles are still present on opposite poles of the cells. So then here, let's have our centrioles. Okay, so these are the centriole. But now the spindle fibers have grown and developed and attached to the centromeres of the chromosome. So draw the spindle fibers in a way where it's showing that they are attaching to the middle of the chromosomes like this. Okay, so this would be our spindle fiber. Okay, so then let's move to the next phase. Before that, let's review it. So we said during the metaphase, we have the chromosome aligned in the mid line of the cell, which is called metaphase plate. So remember, metaphase starts with M and middle starts with M. So the chromosome lined up in the middle of the cell. And then you saw how the spindle fibers attach it to the chromosome. And now let's move to the next phase, which is called the anaphase. In anaphase, the centrioles are still present and opposite end of the cell, but this is where the centromere divides and the sister chromatids separate. So we draw four spindle fibers on each side and draw the sister chromatid as a single strand that you want to show they are being separated and pulled apart. So as you can see, now we draw the cell as a little bit like stretched or kind of the oval shape because that is the time that the cell is being to stretch. So then let me show the centrioles first. So here we have to have our centriole at the opposite poles of the cell. Okay, so that is our centriole. And then let's show the spindle fiber as we have four of them, four chromosomes. Let's draw the four spindle fiber and then pay attention. Right now, we are dividing, separating the sister chromatid from each other or dividing the centromere. Do you remember we said for chromosome, we are going to define the centromere, the part that is going to connect these two sister chromatid. Each of these is called sister chromatid, right? Okay, sister chromatid. So at this time, we are going to separate the sister chromatid and move them toward the opposite pole. So then each spindle fiber should be attached with one of these sister chromatids, something like that. Okay, now each sister chromatid called chromosome. Then we have the last phase of the meiosis. It's called telophase. Let's draw a cell as peanut shape. This is because the cell is beginning to pinch in for the final separation. In telophase, the spindle fiber disappear. The centrioles are still at opposite poles. So then let's draw something like that, as we still can see the centriole at opposite pole. Okay. And then the good news is that the nuclear membrane reforms, or we can say the new nuclear membrane forms. So then now we are going to have our new nuclear membrane, and then around each set of chromosomes. So we should have 1, 2, 3, 4, 1, 2, 3, 4 of the chromosome. So we can show this with a, as you can see, the dashed line. So let's label this as nuclear envelope. And then we have 4 chromosome on each side. So because we have 2 new identical cells that are forming each with a full copy of the DNA. So then we have 4 chromosome here. So let's see. We know that telophase is the last phase of mitosis, right? So chromosome becomes indistinct chromatin again. So then gradually we are actually not seeing the chromosome as visible like we had in the prophase. We saw that, oh, there is no spindle fibers anymore. And then we created the nuclear envelope one more time, and then nucleolus, which is these little dots. We also can see this inside of the nucleus. And then after mitosis end, we have cytokinesis. So cytokinesis is a phase after mitosis. So after mitosis, when we divided the nucleus in two, we are going to divide the cytoplasm and organelles. So cytokinesis is the last phase of cell division. But pay attention, it's not the last phase of mitosis because we divided the mitosis into 4 phases: prophase, metaphase, anaphase, and telophase. And cytokinesis happening after mitosis when we are going to divide the cytoplasm and organelles. Okay, let's finish this video with question to see if you can remember what we talked about this so far. If a human cell with 46 chromosome undergoes mitosis, how many chromosomes will each cell have? We learned that during the mitosis, we should have the identical cell, daughter cell as their parent cell, right? So they're genetically identical and also they should have the same size as well. So when the cell with 46 chromosome undergoes mitosis to increase the number of cells for growth, for repair, for replacement, nothing gonna happen in the like number of the chromosome. So after division, we are going to have two daughter cells which each has the 46 chromosome. Okay, thank you so much for watching.